warm welcome back to another episode of Science Changing Life. My voice may sound a little different to the regular listeners out there. I'm your guest host, Lauren Fish. Today, we're learning all about the mitochondria. While you might remember them as the powerhouses of the cell from your high school biology class, it turns out they're a lot more complicated than that. Joining us is Danielle Grotjahn, who is studying mitochondria to reveal how they impact everything from cell stress to disease. First, Danielle shares what inspired her to pursue this type of research and how a microscope changed the trajectory of her life.
There wasn't any moment when I was younger that I sort of thought, I want to be a professor or a scientist, but my scientific curiosity was always a common aspect of my childhood. And I think for me, the initial curiosity was just growing up, being younger. I grew up in Wisconsin, playing outside in the woods. My grandparents had like 5 acres of land with streams and gardens. And so just really became fascinated with, I guess, just life.
Yeah, the natural world.
The natural world, exactly, yeah. And then, it's a very cheesy story, but it's true. When I was 9 years old, I was gifted for my birthday a microscope kit, like a beginner's microscope kit. And I remember thinking it was so cool. It looked, like something a real scientist would have.
Did it come with a lab coat too? Or?
It did not come with a lab coat, but it came with like the little glass slides and like all the oil.
You could plate everything. And,
yeah. And so, I would go around and I found it really fascinating to be able to take leaves or like scrape the inside of my cheek and look at it. And it was sort of like unlocking an entire new microscopic world. I'd been exploring the world around me, but it was now a new world I could explore.
Yeah, absolutely. I feel like I can exactly picture the microscope you were talking about. I feel like every kid in that age range, like of that era, had one of those.
Totally, yeah. And there's something about when you're younger having like an adult thing,
Yeah.
that you just...
Oh, this is so formal. I'm sure your parents are very proud of the progression. I'm sure they're very thankful that they purchased that.
Yes, absolutely. Yeah, they can take the credit for that one.
Yeah, that's awesome. So with that covered, hoping you could tell me more about your scientific background, what led you to joining Scripps Research.
Oh, yeah. So as an undergraduate, I enrolled in what's called this Honors and Research program. And it was actually a great program. It was at the University of Wisconsin-Madison, where I did my undergrad. And it basically mirrors on some level what you can expect your trajectory to be in graduate school. So Your first year, you do kind of like rotations, you identify a lab. Second year, you write a proposal, and then third and fourth year, you work on that undergraduate research project. Yeah.
That's so
it's
cool
incredible
because
program.
I feel like undergraduate so rarely kind of mirrors the graduate experience. So you would know exactly what you'd be getting yourself into, especially with something like science where you probably knew you wanted to eventually go to graduate school.
Although I can say I don't think anything truly prepares you. I mean, like on paper, it mirrors it.
You're
but
like, this makes sense.
Yeah. But, going to grad school was a different beast, I would say. But yeah, so my undergrad research was focused. It was actually very different from what I did in graduate school. So I was working in a developmental genetics lab.
Okay.
And we used these tiny fish as a model organism. They're called zebrafish. Mm-hmm. So my project basically involved collecting freshly fertilized zebrafish eggs. And then I would immediately collect them and then take them over to the microscope and then watch them literally life grow in front of me. Wow. So I'd watch them go from like a single cell, and then it would divide to 2 to 4 to 8, continue on.
Full mitotic process.
Yes, exactly. My project was trying to look at how they call it the spindle apparatus. It's components that help basically pull apart the DNA and then also the cells divide, how the orientation of that spindle apparatus affected basically how the cell division process occurred. Mm-hmm. Yeah, so I was basically just watching these spindles go and calculating then with the images, the orientation and the cell shape. And
wow.
Yeah. So it was like, I think that for me was really crystallized the idea of using microscopy and sort of having this visual representation of life, mm-hmm. and then transforming that into quantifications or numbers that we can then understand how life works.
Yeah.
Yeah. So
and like when something might go wrong,
yeah.
or kind of all of the nuances involved in that.
Exactly. Yeah. And then I spent a lot of time on a different type of microscope, like a confocal fluorescence microscope, they call it. And it's very powerful. You can look at things in real time. You can look at all these dynamics. And then very naively, I think as I was thinking about going to grad school, I was like, what are other types of microscopes out there?
Yeah.
And so, I found out about this like electron microscope. So that sounds pretty cool. And then, somewhere in my Google searches, I came around this term cryo-electron microscopy. And I was like, that sounds fancy. And so, I think then I just, Googled cryo-EM, cryo-electron microscopy, and graduate programs. And then Scripps came up as like the top one.
Yeah.
So that's what led me to Scripps, was honestly just that like naive curiosity of like following this like passion. And then, of course, Scripps is a top graduate program. So that really fit in nicely with sort of my ambitious goals of what I wanted to get out of grad school.
Yeah.
So that's what led me here. So I made the move. I had lived in Wisconsin my whole life. So this was my big...
I was going to say must have been a big shift.
Yeah.
Coming out to the West Coast and San Diego. Yeah.
So it's like, I'm very happy here. And I think that's, probably, there's lots of reasons why I'm still stayed here, but kind of the scientific environment is a huge component of that.
Yeah, absolutely. And just being surrounded by it, especially up here, like on the Mesa and everything.
Yeah, absolutely.
And then I do have to ask, is the mitochondria the powerhouse of the cell? I think all listeners will want to know the answer to that question.
So mitochondria are sort of the main muse or inspiration of my laboratory's research. And out of all of the cellular components, all of what we call these organelles, they by far have the best marketing team because they came up with this catchphrase that is burned into everyone's brain.
Infiltrated everyone,
Yeah, it's almost like a cultural phenomenon at this
point.
So. There are memes on the internet like, I didn't learn how to do my taxes in high school, but I know the mitochondria is the powerhouse.
And you're like, that's more important.
Yes. Yeah, so it's actually like, it's a pretty powerful tool, I find, to communicate or get people interested, maybe that are not scientists in my research, because you say mitochondria and or you say powerhouse of the cell, it immediately triggers something.
Oh, yeah, I remember that from freshman year biology,
Yeah. But I have to say, if I were on the marketing and rebranding team for mitochondria, I might switch up their catchphrase a little bit. So we call them the powerhouse of the cell because they generate energy, the currency, the cellular energy currency called ATP. But we now appreciate that they do a lot more for the cell. And they're now sort of, people are changing, not just me, but others are sort of changing their catchphrase to something like the stress sensor or the signaling hub, or I think I even saw someone call them like the CEO
I have not heard that one.
Yeah. That is not my original. I wish I knew who to give credit to on that one.
Someone else on the marketing team.
Someone else on the marketing team. Yeah. So what's really fascinating about them is what we're starting to appreciate is that they're an essential hub to receive different signaling inputs from the cell, and then they make these critical decisions of how the cell should respond. And so, for example, they have a huge role in dictating cell fate. So whether a cell activates pathways to stay alive and keep growing and propagating, or whether a cell initiates programmed cell death, what we call apoptosis. And what my lab is really interested in is understanding how that happens. And in particular, what's even cooler about mitochondria is that a lot of its ability to respond to different stress signaling pathways has to do with its ability to change shape. So they're like these like shape-shifting entities inside the cell.
Yeah.
So they can divide, they can fuse together, they can divide just parts of them. Like if they have a damaged part, they can selectively get rid of that. They can be transported all over. So they're they're very dynamic.
Absolutely.
Which is like in contrast to kind of how you see them in a textbook. Usually
it's like rigid structure.
Yeah.
Powerhouse, you think, not really moving and just kind of like
just like chugging
a foundation. Yeah, exactly.
Yeah. So my lab is interested in studying basically the interactions that help them change shape in response to these different integration of these stress signaling pathways. And we are harnessing a cutting edge imaging technology to do this. So, mitochondria have really been an inspiration for microscopists for decades. A very famous cell biologist, Palade, he was the first to really image a lot and give us a view of the inside of cells and describe these entities that make protein called ribosomes in the cells. And he also has these very famous, beautiful images of mitochondria. And so throughout the last century, microscopists have not only looked at their structure and how they're shaped, but how they move, all of these dynamics. But the one thing that we've lacked is really an ability to look at the very fine details of them. So we can kind of see, it's like being able to look at a car and you see it move, but you don't really understand what are the parts that make it
allowing it to do that.
Exactly. And so traditionally what's been done is, like, if we're going with this car metaphor, we can take apart the car and then we can look at the fine details. So you could maybe like take out the spark plugs.
The engine. My knowledge of cars.
I know. I was like, I really backed myself in the hole with this one. So we could see that, but we haven't been able to see, the detailed spark plug or engine architecture in the context of the overall car. And this cryo-electron microscopy, or this cryo-EM that I do, it enables us to look at the entire car, the entire mitochondria, and all the detailed parts that make up the mitochondria itself.
Cool. When you're talking about the cell death component, I remember one of my favorite quotes from my cell biology teacher in college was talking about your cells are in a constant battle if they should live or die.
Yep.
And my mind was just blown at that. I'm like, what? That's crazy.
I know. Yeah, I find you can't think too hard about, like, I had that before too. I was like, when you think about, like, I have to stop myself from being like, there are mitochondria doing all these things in my body. Like, it just freaks me out.
You're like, no, I don't have any mitochondria.
Yeah.
It's like completely separating yourself from that.
Yeah. No, it's totally fascinating, and I think even though they're very widely studied, there's still so much we don't know about them, and they're very complex. And so it's really exciting for my group, because when you're sort of the first to look at something in a completely new way, which I think is really what fuels my passion and curiosity for science.
Yeah, and it goes back to what you're doing in your undergraduate days too, really taking those very fine details to kind of unlock whether it's, that's something about disease or just the mechanism in general.
Yeah, and for me, I've always been a visual learner, so it just makes sense to approach biology, research, and science from that first aspect of, I just want to see it. I want to look at it from every angle. Yeah, and I think that's also, anyone who's sort of worked with me knows that my go-to when I'm trying to understand information is to go up. We have a whiteboard in lab. We have several, actually, but to just go up to the whiteboard and start drawing things or on a piece of paper, because that's really how I take in information and understand it. And so I think that that's very much in line with how I approach my scientific research as well.
So what is the biggest question in this area you're hoping to answer, the biggest obstacle you're hoping to overcome as you're studying these mitochondria at that very fine level?
Yeah, I think just going back to this idea of looking at the detailed structure of the parts, and as well as having the context of the whole, so looking at the parts of the car and the whole car. So I think one of the challenges that we have with the type of microscopy we do, so I've talked a lot about cryo-electron microscopy, or cryo-EM. There's a specific type that my lab specializes in, which is called tomography. It's like taking a microscopic CT scan of your sample, and in that you have this reconstructed representation of what the inside of the cell looks like. And part of the reason why we haven't been able to do this for, until recently is because we've had this barrier in that it turns out even though cells are very small, when it comes to some of these microscopes we're using, they're too big to really penetrate and see all of the detailed information. That's been a huge problem. In the last 5 to 8 years, other groups have really harnessed technology to allow us to carve into cells and to trim them so that they're thin enough that we're able to actually visualize these fine details. And so I think what's really exciting for labs like mine that are just starting out, my lab is about 3 years in, and we have this really unique expertise. When we started off, we were one of the few labs that's really specializes in this carving of cells and imaging, CT scanning of cells. Yeah. And so the challenge with that is that sometimes you want to go after a really exciting question. For example, how do mitochondria change shape? What are the components of mitochondria that allow them to change shape? You start with this very ambitious question, you start tackling it, and then you realize there's some sort of roadblock, like technology roadblock that you may encounter. And when you're on the bleeding edge of a technology, you may look around and realize that no one has solved this problem before. And so you can't just use something that someone else has done. You actually have to come up with this. You need to invent it. Yes, exactly. There's a plethora of questions that we're trying to go after in terms of what is the machinery that helps mitochondria divide? How do these components assemble around mitochondria and really impart force to divide mitochondria membranes? We also want to understand how not only the mitochondria, but other components inside the cell, like other organelles. There's another really fascinating organelle. I mean, mitochondria is my favorite, but my second favorite.
I was going to ask if mitochondria was your favorite.
They're probably my favorite. One. Yeah, I have to say, I do have a love for another cellular component, which are called the cytoskeleton, which are those like spindle.
The little like microfilaments.
Yeah, exactly. Yeah. So, yeah, it's hard. Don't make me choose. Yeah.
I won't tell anyone.
Luckily, they interact a lot, so I can look at both of them at the same time.
You don't have to make favorites underneath the microscope.
Yeah, so we want to be able to look at how these come together, but oftentimes it's just we hit a wall, and so we have to kind of take a break from addressing that specific question to kind of figure out on the technology side, how are we going to overcome this barrier? And I think that's, one of the exciting but also challenging aspects of being, part of any sort of advancing or developing approach.
Yeah, being a pioneer at the forefront of something.
Yeah. So it really feels, though, like my lab is at this perfect, like we're just at the dawn of this era where we've spent a lot of time and hard work. I mean, I have an incredible team of trainees, postdocs, graduate students, technicians who have spent a lot of work implementing, all the latest advancements, and we've developed our own approaches. And now it's like this perfect synergy of just being able to start to tackle some of these questions that, I set out wanting to tackle at the beginning, and now we're finally able to do it.
Had some technology roadblocks initially.
Yeah, exactly.
But that's so incredible that you are really at the forefront of this, and that you're really changing the game, not just in, studying mitochondria, but I'm sure the rest of the cell organelles as well, and how everything interacts.
Yeah, I mean, I think there's, before I started to be a group leader, I think I had this fear, like, I'm going to run out of ideas or, but actually it's the opposite.
There's too many questions.
There's too many questions, and you have to pare it down. And the biggest challenge now is, kind of prioritizing or focusing and figuring out what are the interesting questions that you want to go after. Because there's not there's certainly
there's a long list.
Long list. Yeah, exactly.
Well, and what you were saying earlier about how in science you have to get more narrow, right? You have to kind of focus on something. But I also feel like the more narrow you get and the deeper you go, the more questions you have, because biology can always get that level deeper. And more complicated. Yeah. And so, I'm sure the questions just keep coming as you're starting to learn more.
Yep, absolutely. And that's what's really great about being at Scripps, and also I think Scripps has several other faculty that are similar to me, where they have this unique tool or approach, and then it really lends itself well to collaboration. So, in my lab, we're really focused on mitochondria and harnessing this imaging technology to look at mitochondria. But I can sort of feed that other thirst for looking at other parts of the cell by collaborating with other labs that are focused on other parts of the cell. And in that sense, it really gives a great back and forth scientifically for making new discoveries.
Right. And I think everyone I've talked to at Scripps has just really echoed that collaboration is inherent in every lab here. And, you're at the, cross-section of so many different disciplines, like structural biology, molecular biology, and just to be able to have, access to like other people and people willing to talk and collaborate openly, I think is definitely something really unique to the culture here.
Yeah, no, absolutely. And I think the fact that I can talk to not only people who specialize in areas different from mine, but like the top researchers in the field that are different than mine, in chemistry and chemical biology and neuroscience. I mean, it really is, it makes it incredible that you can interface with, those that are really on top of the game in that aspect and learn from them and just try to understand what makes them curious about science and how maybe what we're doing in my lab might be able to help them illuminate new views of what they what they're interested in studying.
Or if they're asking similar questions that you're asking and what they've learned from their own discovery. So we've talked a lot about the form of mitochondria and the structure of mitochondria, but how have you found that mitochondria implicate things like disease or kind of the inner workings of the cell?
Totally. I mean, we would probably need like 2 hours to go over all of it. It turns out mitochondria are implicated in so many aspects of cell health, and because of that, also in disease. Just to narrow down the things that my lab is interested in. One, as I mentioned before, there's this cell fate aspect about mitochondria. And so that's really important for diseases such as neurodegeneration, where you have mitochondria thought to play a role in the neuronal cell death process. And so a lot of neurodegeneration, at the heart of it, is overactivation of pathways that lead to premature neuronal cell death. On the flip side, you have diseases like cancer, where the pathology or the detrimental aspects of cancer are that you have uncontrolled cell growth. So you have these two sort of very groups of devastating diseases where one of them, on the one hand, you have where you want to preserve the life of cells, and on the other, you want to control the division and sort of control that programmed cell death aspect. And mitochondria are sort of play a role in both.
Right.
But we don't quite understand how their function ultimately leads to these very distinct outcomes. And one thing that's similar is if you look inside of, for example, cancer cells, and if you look inside of, for example, tissue from a patient that suffered a neurodegenerative disease such as Alzheimer's, what you find in both of them is that the mitochondria are very fragmented. They're very small. So there's like this hyperactivation of the division, or we call it the fission pathway. So that's confusing.
Right, because they are two different fates, two different outcomes.
Two different outcomes. And so something must be very different about the interactions that mediate the division of mitochondria where in cancer as opposed to a neurodegeneration. And so, so that's interesting aspect number one. But it turns out that even in just a normal healthy cell, mitochondria are constantly dividing, fusing. I mean, and that's just a normal part. So it's not like dividing mitochondria automatically equals disease. I mean, that's a part of their function.
Right.
And it turns out we don't even really understand how mitochondria divide, even in a healthy situation, even in a normal functioning cell. And so what my lab is interested in doing is looking at all these different contexts, both in healthy states as well as in, for example, patient-derived cell lines or different models of disease in cell culture to understand what is this machinery that helps mitochondria divide. And by understanding what it looks like, how it assembles, how everything interacts on that very detailed level, both in healthy samples as well in these two very distinct disease states, we might be able to understand what areas we can eventually target or to halt the progression of this from happening.
And just better understand.
Yeah.
That's really interesting that, just comparing it to normal cell division. We know so much about the cell division mitotic process, but why has understanding mitochondria division been such a barrier? Is it because of the technology kind of dearth of,
That's a great question. I should say, we do know quite a bit about it. We know a lot about what proteins or what these molecular players are involved. We know that there's this endoplasmic reticulum, I guess my third favorite organelle. We know that that's involved. We know my second favorite, the filaments are involved. But we don't really quite understand how they assemble. And getting to your point, it's really because we haven't had a way to dive inside of cells and look at it with enough detail to be able to understand those interactions.
Right.
Part of the problem, too, a lot of times in biology, in order to understand the details of something, we sort of take all the components out of their cellular environment and we look at them in isolation. But it turns out with mitochondria, they rely so much on their interactions with everything in the cell.
In the environment.
Yeah, that you can't really do that. It's like observing a lion in captivity versus out, in its environment.
In its natural habitat, right?
Yeah. So that's been a huge challenge. And so that's really why a lot of these questions have been left unanswered.
So when you're not in the lab, what are some of your other hobbies or interests?
That is a PI's favorite question. I think as a graduate student, I got really, and also just living in San Diego, it's hard not to become very active when you're
I think you have to. It's just part of the culture.
Yeah, like if you're sitting on your couch and it's just sunny outside, it's like passive aggressively sunny,
Yep.
You're like, I gotta get out. And so, and for me, sort of working out and exercising became a huge stress reliever as a graduate student. And so, I think I carry that with me now. I love to be active. I do lots of different workouts. Like I love yoga. I do the spinning, indoor cycling. And, beyond that, just going to the beach. Also, I recently purchased a paddleboard, so I've been going on that. Yeah, so I think for me, that's probably outside of work. Like, if there's an activity I'm going to choose to do, it's probably something either outdoors or something very active.
So final kind of round-up question.
Okay.
If you could give one piece of advice or wisdom to an up-and-coming scientist, what would it be and why?
So I think probably the best piece of advice I could give is to, as early on as you can, really identify, recognize, and then eventually celebrate both your strengths as well as your weaknesses. And I think this is something that, I take very seriously because for a long time, I never really felt like I identified as like a scientist. Although, I pretty much excelled in my biology courses in high school and in college, and, I was in a lab, but I sort of felt like there was some part of my personality that didn't fit, the quote unquote mold.
Scientist mold, right.
Exactly. And, it didn't really faze me too much, but it was always kind of this lingering thing. And I couldn't quite put my finger on it, though, because it's, like I said, I was in the same study groups as everyone else. I was doing the same things and my same cohort of graduate students and whatnot. And I actually, I took sort of a career planning course as a graduate student. And part of that, they have you go through the sort of battery of these different personality and strengths finder tests. So I did like the Myers-Briggs, strengths finder, all of that. And that was super illuminating for me because, you kind of go through it and you can identify those parts of you and you read it and it's like, wow, this is me. This is me. Like, this is like crazy. And it's a spectrum. So there's some things you're kind of like, I don't know. But there's, you cling on to those things that you really identify with. And then on top of that, being able to really identify what your strengths are, like what your natural sort of inclinations are. What do you everyone's born with like certain natural abilities. Doesn't mean that you can't develop, skills in other areas you weren't born in, but you have this natural tendency. And part of this, I think, was also they had this list of what would be the
ideal career based on your personality.
And I think scientist was like number 8 or number 10 for me. And at the top was like social worker, teacher, lots of other sort of things. And, I think that actually I wasn't discouraged by it. It was like an aha moment. I was like, oh, wow, this is actually really kind of crystallizes this feeling that I've had. And it's not like there's something wrong with me, or most importantly, it's not that I don't belong here. I've made it here. I'm here already. Right. It's that this can kind of explain like my natural inclinations towards certain things is different than maybe others. And I think, it's not just science, but I think science definitely on certain aspects, it selects for certain personalities over others. And it doesn't mean that if you have a different personality, you can't be a scientist.
Absolutely.
Yeah.
Well, and you can, like you mentioned teaching, that's such a huge part of your work here and leading a lab.
So I think for me that's exactly what happened. It was that moment where I realized and I could focus on that and think, what is it about the things that I'm naturally good at that I can bring and actually enhance the scientific community? And on top of that, it also helped me understand areas that I struggled with. Why was, in certain ways that like we were talking about, I'm a visual learner, that there's a lot about how you perceive and input and output information. And I think for me, it was also very useful to understand what areas I sort of don't naturally gravitate to, and what I can do for myself to sort of compensate and overcome for that. Right. And so I think that's really important because there's this concept that can happen called imposter syndrome, where it's exactly what I feel like I don't belong, like I'm going to be found out. And so for me, I think this, as a group leader now, it's really something that I value and place a lot of weight on, is really creating in my lab a very diverse environment that spans different personalities, different demographics, socioeconomic statuses, all of these across the spectrum of diversity, because I think it's important to have not only all different voices, but to think about how the labs can better represent the communities we hope to serve.
Absolutely.
And so, I think that would be my advice is, if you're feeling sort of, insecure or not that you don't fit in, try to understand yourself a little bit better and contextualize and identify what you're really good at and how you can harness that and bring it to the table.
And with that sage advice, that brings us to a wrap for today. Many thanks to Danielle for hopping on and giving us a much better understanding of the different roles that the mitochondria play in the cell, and especially how these organelles can impact diseases like cancer and neurodegenerative disorders. To learn more about Danielle's research and other Scripps Research content, be sure to check out the show notes. A big thank you for tuning in with us today on another episode of Science Changing Life. Until next time.